Okay. One second.
Okay.
Um, can I start presenting now? Okay. Well, thank you for confirming. So, magandang hapon. Good afternoon, everyone. I am Jason Tuang-tuang, a PhD candidate from the Centro de Investigaciones Biológicas del Noroeste in Mexico. In fact, I am on my last few months of my PhD program. And on behalf of my um, directors, it is a great pride and honor to present this as one of the significant outputs of my PhD project entitled Micro-morphological variation of Pocillopora grandis is associated to temporal changes of corallite development. So to understand and protect coral reefs, we first need to know which corals are present in a certain coral reef or coral community. And traditionally, coral identification and reef monitoring heavily relied on the skeletal architecture or the morphology, including, the colony shape and the shape of the branches representing macromorphology. And also the corallite features serves as micro morphology. And these traits help us not only to identify species correctly, but also to track changes in the reef communities over time. And in order to track these changes, several studies have performed morphometric strategies and techniques to different coral species, as you can see here in the slides, in some species of Borites and Pocillopora. So they have made some several measurements in the macro morphological and micro-morphological level or at the corallite level. So but first, I need to identify what are corallites. So, corallites are actually the core skeletal unit in scleractinian or hard corals. So, these are examples of corallites of Pocillopora. And as you can see, they support and provide shelter of the coral polyps, as you can see in the photo, and they have different microstructures with different functions. For example, the columella, this central projection, which supports the coral polyp, and these appendages or like blade-like structures are called septa, which aids in the digestion by expanding the polyp's stomach. So these micro-morphological traits not only determine or support the colony function in total, but also used for taxonomic diagnoses, particularly in highly plastic coral Pocillopora. And Pocillopora are widely distributed throughout the Indo-Pacific region. In fact, in some of the areas of the Pacific, they are the main reef builders. And we focused our study in the species Pocillopora grandis, and in the Western tropical Pacific, where I'm originally from, it is easily identified with a lamellar or a columnar thick branches, and its corallite possess a styloform columella and a rudimentary septa. However, in the Gulf of California or in the whole Eastern tropical Pacific, as you can see here in the photos, Pocillopora grandis species tend to have different morphologies that match with the other Pocillopora species in worldwide. So, despite genetically confirmed that these colonies belong to Pocillopora grandis based on uh, mitochondrial lineage using ORF and control region markers. So, why? So, one of the backgrounds of these studies that these differences in the morphology were actually responses to different hydrodynamic regimes where the coral colonies are inhabiting. For example, thicker branches, narrower distances between fragments can be expected to coral colonies in high flow condition compared to the ones in low flow with thinner and wider branches. Thus, morphological plasticity is actually apparent. So this led us to the question, If macromorphology in Pocillopora can shift in response to different hydrodynamic regimes, do micromorphological features remain stable at species level traits, or do they also vary with hydrodynamic environment and or other factors? So to respond to that research question, so we conducted a study at two Pocillopora dominated coral communities with contrasting hydrodynamic regimes in the Gulf of California. And we focused our study in one of its morphotypes, the morphotype inflata, which can easily be identified with its swollen branches. And these colonies have been studied since 2011. So this will also, like, we come up to decide to study this one to minimize also the variation and maximize the chances of observing morphological changes at the corallite level. So we collected coral fragments and around around 10 centimeter from eight uh, different matured colonies at each site in low flow and high flow, and around 8 to 10 meters apart of of these colonies, so that to avoid that we are resampling clones. We're sampling clones, and these colonies have been resampled in four sampling periods in March 2023 to November of 2023 and April of 2024. And these samples were then preserved, and but we only used the around 2 centimeter size that was cut in the middle section for processing and imaging using the scanning electron microscope. And once we generated, for example, this kind of image, so all of the corallites were examined to assess and classify them and classify their morphological features. So as a result, it's a mess, to be honest. So the images revealed a temporal variation. So from March to 2023 to April 2024, as you can see here, they reveal, a temporal variation in the micromorphological features. So as what I have said, we have repeatedly resampled the same colonies all over the collection time points, and these micromorphological traits do not completely align with Pocillopora grandis, but they align with other Pocillopora species that cannot be, that are found in other areas of the Pacific region. So, after careful examination of the images, we actually found out that these changes are developmental stages of the corallite. So here we proposed five developmental stages to describe these growth patterns. So from stage 1 to stage 5. So this sequential variation suggests that corallite ontogeny influences variation in micromorphological traits as opposed to plastic responses uh, shaping all correlates at once. And of the micromorphological traits, what we have seen observed is that the columella and also the septa vary the most. And as what I have discussed in my previous slides, that these are used for species identification for Pocillopora. In fact, from stage 2A to stage 2 5, they belong to other species. But that's what I've said. We have recorded them here, and we have monitored them to how frequent these stages. So all correlate stages were observed in each monitoring period, but the distribution of these correlate stages varied across sites. And I just want to highlight your attention to this stage 2A and 2B. So it seems like stage 2A with a flat columella and 2B with a hollow columella, because in uh, low flow site, we have seen a high percentage of recurrence of stage 2A compared to uh, in low flow site compared in high flow, and also a high percentage of stage 2B, a hollow columella, compared with the rest of the monitoring periods. So this creates a huge question for us, to be honest, when we have encountered this one. So honestly, we don't have a direct answer yet. We're still um, trying to respond to this question. However, we associated the highest incidence of these two stages at low flow site with the peak reproductive season for these species, where during the summer season, particularly in September of every year, almost like around 75% of the colonies are reproductively active. So we treat that this flat and hollow columella may provide extra space for developing and mature gametes, especially that this species is a hermaphrodite. And it's also been confirmed in other species of Pocillopora that physiological trade-offs is apparent between reproductive capacity and skeletal growth. And energy when there's a reproduction, it's energetically taxing. So yeah. So, as a conclusion and some takeaways, interest-specific temporal changes of corallite structure were associated to growth patterns and not plasticity. And micro-morphological trait changes could confuse species diagnoses. And growth patterns apparently tied to seasonal changes in the Gulf of California, and seasonal reproduction correlates micro-morphological changes in these species. So I just want to thank the people in Marine Biogenomics Lab at CIBNOR, and of course the Ministry of Science and Technology, also my research center, the lab of my director at Louisiana State University, and the Bay Lab at UC Davis, and of course SSE for this opportunity to share this part of my project. And maraming salamat po. Thank you so much, everyone. Muchísimas gracias. And I'm ready to answer your question if there.
Question? Put your hand up if you do. Well, I have a question.
Okay.
Have you done DNA barcoding on this coral to prove that this is the species you think it is? Because I agree, it does seem like it's all attached with one another, right?
Yes. Um, this is the was this these colonies have been identified already or genetically confirmed their species since 2011. So we have
Yes.
just this is just like continuation of the project, like of how plastic they are, and just trying to go into the micro-morphological level. In fact, I have, this is just a part of the study. We did a gene expression analysis for this one, and even, we used the transcriptome, to further confirm, the species. It's really Pocillopora grandis, even though
The micromorphology doesn't look like that. No, that's a really good point. It's nice to have that data behind you when you're trying
yeah.
to publish
So
it, and people say, "Oh, maybe it's not that species." You say, "Oh, yes, it is." Very,
yes.
very good. Any more questions? I think we still have a couple minutes.
Yeah, question. Um, were you surprised that the micro-morphological properties didn't allow you to better identify were there I'm not that familiar with, the marine system. Were there other systems where this had worked well?
Yeah, yeah. No, 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 no worries. Actually, when I started my PhD, I was only focusing on the macromorphology, but one of the groups in the Tara Pacific published a paper um, that some species in the Western Pacific, that their features, did not actually align. So I was kind of expecting that it might not, but the thing is that I thought that in every time point, because we have the samples, we have collected samples. So I thought that every time point, we might just gonna be able to encounter just one trade. But in every season time point, it's like, why all of a sudden that they're different? in every tampon. So that's why after, you know, actually we have like around, I don't know how many images that we have, but yeah, it just, we have, well, at the end of the day, we just like, it's like a jigsaw puzzle. That's why we came up with those, with those developmental sequence of the corallites. So thank you.
So we have to move on to our next speaker. Corals were very nice, but now we're going to learn about organic agriculture and weeds. So Robin, take it away.
All right. Yeah, so I'll be talking about plasticity and evolution in weed adaptation to organic agriculture. I just finished up my PhD working with my advisor, Jeff Connor, at Michigan State University's Kellogg Biological Station. All right, so why am I interested in studying weed ecology and evolution? One reason I think this work is pretty critical is that weeds threaten our global food production, but we know that current control methods relying on herbicide applications to genetically modified crops are financially and environmentally unsustainable. So while herbicides are a super useful tool for weed management, they have the drawback that they have a substantial carbon footprint and they can negatively affect off-target organisms. So we really need some biologically informed solutions. From the more fundamental side, weeds can provide useful case studies for understanding rapid adaptation to human-altered environments. And that's because the selection pressures are often strong and applied in many fields across the landscape. So what might some of these selection pressures be? I've grouped some agents of selection in crop fields into 4 categories. First, we have our chemical inputs, herbicides, fertilizers, and most of the prior research on weed adaptation has been in this area. But today I'm going to be examining the organic environment, so non-chemical selective agents, and these are much less well understood. These include things like disturbance, so removal of above ground biomass at harvest, disturbance of below ground biomass from tillage and cultivation, changes in the frequency or abundance of biotic factors like pollinators, herbivores, and microbes, and changes to abiotic resources like moisture, light, and heat. So overall, it's unclear if weeds from nearby uncultivated areas are pre-adapted to invade organic fields through adaptive plasticity, or whether they evolve in response to selection that they experience in these fields. And the idea of rapid flowering as an adaptation to agriculture has previously been supported by a few prior studies examining trait differences between populations from agricultural versus non-agricultural habitats. And this makes some sense because organic fields are harvested before the winter killing frost in an uncultivated area. Water availability may also generate strong selection on plants in organic fields because compared with uncultivated habitats, they have more bare soil early in the season. So this is just a visual example on the left here comparing an old field and a soy field, and we see a lot more bare soil on the right. And soil disturbances from weeding can further lead to soil drying. And we can see in this plot on the right, we just have our water content near the soil surface, and we see it's lower in the organic field. We set out to answer 3 questions. First, are weed populations from agricultural habitats locally adapted to their home habitat? Are there plastic trait changes between agricultural and non-agricultural habitats? And if so, are they adaptive in the agricultural habitats? And third, have populations from agricultural habitats evolved trait changes compared to those from non-agricultural habitats? To study this, we use 3 annual weedy species long established in North America, with populations both in actively cultivated organic fields and uncultivated habitats such as roadsides, ditches, old fields, that kind of thing. And so we've got ragweed on the left here. If you have allergies, you probably already know about this plant. In the middle is lamb's quarters, and on the right is horseweed. To test these questions, I started by collecting seeds from paired non-agricultural and agricultural sites from each of the 3 species, collected from 3 different regions in Michigan. Then grew all these plants together in a greenhouse common garden to standardize the parental environment and generate seeds for the field experiments. In the first year of the field experiments, we transplanted seeds directly into the field in 2 replicate blocks. Unfortunately, we had no emergence of one species and low emergence rates in the other 2. We think this was partially due to unusually dry conditions that year. And for that reason, in the second year, we instead transplanted seedlings. And this method was really successful. The plants were a lot more competitive with the crops. And here's just a visual example of a ragweed plant at harvest in 2023 versus 2024 on the same scale. And so for that reason, today I'm just going to be focusing in on these 2024 results. So we collected data on the plant's survival to flowering, the fecundity of the survivors, and multiplied that gave us their total fitness. We also looked at traits. So we expected plants in and from agricultural habitats would flower sooner. We then looked at a few different measures related to drought adaptiveness. So we looked at the ratio of leaf mass to leaf area, expecting that plants in and from agricultural habitats would have higher leaf mass per area. We also expected that they may have lower stem to root length ratio so that they can invest more in those root structures to gather water resources, and that they would have higher water use efficiency, which is just the photosynthetic rate divided by water loss. Okay, so going to our first question, let's take a look at some expected results. So here is just plants that are growing in the agricultural growth habitat. And evidence for local adaptation would be if the agricultural populations have higher fitness than the non-agricultural populations. Alternatively, we might find no differences in fitness between populations or local maladaptation, where agricultural populations have lower fitness in their home habitat. So what did we find? So here I'm going to be showing each of the 3 species, ragweed, lamb's quarters, and horseweed, the source population habitat on the x-axis, and estimated total fitness on the y-axis. So we see that for ragweed and lamb's quarters, agricultural populations had higher fitness when growing in the agricultural plots, supporting our local adaptation hypothesis, but a slight marginally significant effect in the opposite direction for horseweed. And to better understand what's driving these patterns, we then took a look at the fitness components of survival and fecundity. And we found that for ragweed, there was no difference in survival, so that total fitness pattern was entirely driven by a fecundity advantage, and similar for lamb's quarters. Horseweed, despite a marginally better survival, it didn't outweigh much lower fecundity. So these total fitness patterns were driven by variation in fecundity. So to conclude for this question, we found evidence that the agricultural populations were adapted to the agricultural habitat for 2 out of 3 of our species. Moving on to question 2. We're looking at some predicted results. So unlike for question 1, we're now looking at traits rather than fitness on the y-axis, and the x-axis shows our 2 growth habitats. The color of the points, red versus blue, indicates the source population habitat. So in this example, there's selection to decrease the trait value in the agricultural habitat, which is matching the direction of plasticity in this first case. We also might find that there's no plasticity in a given trait, or maladaptive plasticity, where the direction of plasticity opposes the direction of selection. Okay, so we've got a bunch of traits, and I'm just going to summarize them here, where we have our 4 traits on the left, the a priori predictions that I talked about in the beginning, and then I'll be showing just the direction of the change. And I'm going to hone in on a few takeaways because it's a lot to look at. So first, we found significant plasticity for almost all of these cases. But if we color in the boxes by whether they matched or didn't match our a priori predictions, we found that most of the time they didn't match our predictions. And then if we colored them by whether they matched the direction of selection, even fewer cases matched the direction of selection. So overall, we found that there's a lot of maladaptive plasticity and little to no adaptive plasticity. Okay, on to question 3. Have populations from agricultural habitats evolved trait changes compared to those from non-agricultural habitats? And if so, are they adaptive in agricultural habitats? As in question 1, we're looking just in the agricultural field growth habitat and comparing populations sourced from non-agricultural with populations sourced from agricultural populations. So in this case, evidence for adaptive evolution would be if the direction of evolution is in the same direction as selection, and we might also find no evolution or maladaptive evolution. So back to our table, but this time looking at evolution, we found significant evolution in about half of the cases, but it often varied by our regional pair. We then found that evolution matched our a priori predictions about half of the time and matched the direction of selection in the agricultural habitat about a third of the time. To get a better understanding of the results, we're going to focus in on flowering time, which showed the largest magnitude of evolutionary change. And because of significant interactions between source habitat and regional pair, these plots now show region on the x-axis, and within each region, the blue dot is the mean for plants sourced from non-agricultural habitats, the red dot plants sourced from agricultural habitats. The black arrows are just showing the direction of evolution. We can then look at how the evolutionary change compares with the direction of selection shown in this red arrow, and we had an intermediate optimum in this case. So overall, we're finding slower flowering among agricultural populations in ragweed, which match the direction of selection, and an intermediate optimum in lamb's quarters, which seem to be only adaptive in one of our regional pairs. In horseweed, there is no significant selection on this trait. So, conclusions from this work. First, when we looked for evidence of adaptation, we found that ragweed and lamb's quarters plants from organic fields were locally adapted to organic agricultural habitats. It's unclear why horseweed plants were not locally adapted. It's possible that in years where survival is the primary determinant of total fitness, we might have seen some evidence for adaptation of horseweed plants. Second, we looked at trait plasticity, and we found that almost all traits were plastic, but these were almost never in the direction of selection, suggesting maladaptive plasticity. And this seems to indicate that plants are either responding inappropriately to one or more environmental cue in the crop field plots, or alternatively that the traits are more important to survival than fecundity because we measured our traits at harvest. We may have missed a portion of the contribution to fitness. Third, we looked at trait evolution, and we found that evolution was less common than plasticity, but more likely to be adaptive. We found large evolutionary changes in flowering time and stem to root length, but they often varied in direction by region, and that indicates either that selection pressures substantially differ in crop fields of different areas, or that genetic differentiation in these traits is due to non-adaptive evolutionary processes like founder effects, and that at least for ragweed and lamb's quarters, evolution in unmeasured traits likely drives their adaptation to agricultural habitats. All right, thank you. I'd like to thank everyone shown on the screen, all these funding sources, and I think I have 1 minute for questions.
If people want to ask questions, would they please unmute and put up your hand?
You're welcome to start sharing your screen, Adrian. And if there's anybody else that has any other questions, you can ask them in the Q&A.
Yes, I would just ask, like, while he's doing that, ask you a quick question. And you said some of the evolution you've observed seems to not be what you would have predicted. It's kind of in a different direction. So you were wondering if it was like not adaptive evolution, right? From drift or something. Can you elaborate on that?
Yeah. So, in some cases, the evolutionary change opposed our prediction, like ragweed flowered slower, even though, we have this strong reason to believe it should flower faster. So I think we're still investigating, I guess, why that might be. But one of the next steps in the project is looking at the genome-wide level and trying to characterize gene flow among these populations to get a better sense for the potential non-adaptive processes. And I'll hopefully be presenting some preliminary results from that at the in-person meeting if anyone is interested.
Oh, very good. And we should probably go on to our next speaker, but please feel free to ask questions in.
Sorry, can I start? Should I start?
Yes, go for it.
Okay, thank you. Well, good afternoon, everyone. Thank you very much for the opportunity also to share my some preliminary data that I have from my dissertation. So in this chapter, I will try to describe some evolutionary characteristics of a highly adaptive weed species in agricultural landscape, such as Lolium multiflorum, or commonly known as Italian ryegrass. So Lolium multiflorum, it in the US serves different purposes. There are production of certified seeds happening in Oregon, and this species is usually implanted as turfgrass, pasture, or cover crop in agricultural landscapes. But at the same time, a feral ecotype has evolved from this species, which is commonly known as Italian ryegrass, which is widely spread across agricultural landscapes in the US and also worldwide. Importantly, this is an obligated outcrossing species system, which enables different populations to exchange genetic backgrounds among them, which might explain their highly adaptation capacity. In agricultural context, worldwide, there are cases of evolved herbicide resistance, and Lolium multiflorum is one of these cases. There are 9 different resistant mechanisms reported in this species that acts against different mode of actions. And within this context, glyphosate, which is one of the main tools to control this weed, has also been affected by the cases of herbicide resistance evolution. So glyphosate is a broad spectrum herbicide. It can control grasses and broadleaves. The mode of action is by blocking the enzyme EPSPS, which synthesize precursors of aromatic amino acids. So despite glyphosate being a highly efficient herbicide and very powerful, plants have evolved different types of resistant mechanisms towards glyphosate. And generally, we term these mechanisms into broad groups: target site and non-target site resistance. When we talk about target site resistance, we talk about cases of mutation at the enzyme level where this drives a alteration in the 3D structure of the enzyme, thus affecting the interaction between the molecule of the herbicide and the enzyme. Or another case is the overexpression of the target site. And this example has been found in populations of Amaranthus palmeri, where ecc-DNA contains the EPSPS sequence, and this drives an overexpression of the enzyme. And another example was found in Kochia scoparia, another weed species important in the US, which has a tandem duplication of the EPSPS. And then on the other hand, we have non-target site resistance, where changes in the enzyme are not the case, but we talk about enhanced physiological processes such as reduced absorption and translocation of the molecule, or vacuolar sequestration, or enhanced metabolism of the molecule. So in the US, cases of glyphosate resistance are a good example of the capacity of adaptation of Lolium multiflorum, as there are different instances of resistance mechanisms in this species. For instance, in California and Alabama, different populations in very distant states, populations with SNPs at the EPSPS level were reported. Then in Arkansas and Oregon and New York, more recently, cases of increased copies of the EPSPS were reported in resistant populations. And in Mississippi, there was a case of reduced absorption and translocation. So due to the different instances of resistance evolution to glyphosate in this species, and due to the different populations and distant states where this happened, our hypothesis proposed that these two types of mechanisms have evolved concomitantly across different populations of Lolium multiflorum, and also that there's different mechanisms of resistance have evolved independently across populations. So to test our hypothesis, we gathered 42 populations from 12 different states of the country. We screened them with high rates of glyphosate of 2,634 grams of active ingredient. We used 36 individuals per population. All individuals were cloned, and the clones were treated in order to later retrieve tissue from the susceptible individuals, which wouldn't survive the treatment. And 27 days after the treatment, we proceed with phenotyping our individuals as resistant or susceptible according to the necrotic levels. After that, we proceed with clean leaf tissue samples from the survivors' clones and also from the untreated mother plant from the susceptible individuals. And we extracted DNA and we sent a collection of 336 individuals between susceptible and resistant to a whole genome resequencing, aiming to a 10x coverage per individual. So with this, we started our analysis by first investigating what type of mechanisms we had in our collection. We first scanned for mutations at the EPSPS level using the software BCF tools and Genius. We also investigated the EPSPS copy number variation using the software MOSDepth, which measures the different rates of coverage variation across the genome. And then we defined the EPSPS copy number as the relation of EPSPS read mean depth and genome-wide mean coverage. We also investigated the genetic profile of our resistant phenotypes. So we conducted a genome-wide study association with two models using the software GMAT in our studio. In the first model, we accounted for population structure by adding the three first PCs plus kinship matrix of genetic relationship. And in the second model, we added the CMV profiles of our individuals in order to find SNPs associated to our phenotypes that are independent from the CMV condition. And then lastly, we perform also selection scans on our associated SNPs or regions of interest between resistant and susceptible individuals to see if there are differential sections of selection across these regions. And also we applied the H1/2 statistics in order to infer if these selective sweeps might be hard or soft sweeps. So in the results, after investigating the mutations, the presence of mutations, we only found two populations carrying mutations at the EPSPS, which were one population from Alabama and another population from California, which were previously reported already. We knew that beforehand, but this confirmed it. And then after investigating the EPSPS copy number, we found that this is the case that mostly drives the resistant mechanism to glyphosate across our collection. We found a minimum mean coverage of 10 at the population SCO1, so Carolina 01, and a maximum of 60 at one of our Oregon populations. And then lastly, and the states where we found this are listed in this column. So then also we found some other populations from Kentucky, Maryland, and Oregon, which did not have any known resistant mechanisms, so we cataloged them as potentially non-target site resistance. Then we proceed with our genotypic association with the two models that I explained before. So after the first model, the GWAS provided us with this figure, with the A figure, where it's presenting a polygenic trait associated to our resistant phenotype. So what we did was we extracted 1 million bases surrounding the lead SNP in each region. We mapped the genes, and then we investigated if there were known resistant mechanisms in these regions, and we didn't find any known resistant mechanisms. So what we did was highlight some potential enzymes that would worth to validate in the future. For instance, we found glucosyl transferase, xyloglucan galactosyl transferase at chromosome 3, which might be involved in glucosylation of xenobiotic enzymes, which is a way of metabolism of herbicides. We found a phosphate translocator at chromosome 4, which might be an active transporter of glyphosate that might be altering the movement of the herbicide. At chromosome 5, we also found another phosphate transporter and also an ABC transporter, which was previously a similar family of the one that we found here was validated already in Echinocloa colonna. So that might worth to investigate later. And then the second model of the GWAS resolved the first polygenic trait into one single locus, as you can see in the B figure at chromosome 4, where the most interesting enzyme that I could find is the phosphate translocator. So that might worth to try at some point to validate. And interestingly, we didn't find any variants at the EPSPS level to be associated with our phenotypes. And then at the selection scans, we investigated first the EPSPS region. The cross-population analysis indicated that EPSPS is under selection in the resistant populations. Then we took the SNP with the highest log p-value, and we conducted a haplotype homozygosity decay analysis. And we can see that the decay is abrupt in this region for both groups, but the integrated haplotype homozygosity indicates that the derived allele in the resistant population has larger blocks than in the susceptible population. And the frequency of the derived alleles in these regions are high in both populations, but slightly higher in the resistant. And then the H12 analysis resulted marginal in this region. And this might be due to the high ratio of H2, H1, indicating that there's a high diversity of haplotypes carrying derived alleles in these regions. And then for the chromosome 4, we had similar results where the lead SNP, the region of the lead SNP of the GWAS is under selection in the resistant population. The IHH indicates that the derived allele it's present in larger blocks than the ancestral allele, which might indicate that the selection on these derived alleles might have happened more recently. Also, the frequency of the derived alleles is substantially higher in the resistant population, than in the susceptible population. And then we also found another marginal H12 in this region that might be again the result of the high haplotype diversity carrying these derived alleles according to the H2H1 ratio. So to conclude, our results are indicating that instances of increased copy numbers of the EPSPS are driving most of the resistant cases in Lolium multiflorum to glyphosate in the country. Also that there is a novel potential non-target site resistant mechanisms across populations that might have evolved recently and as a secondary mechanisms under the CMB. And also that the high haplotype diversity of resistant associated regions could imply instances of repeated evolution of similar mechanisms across our populations. So I would like to acknowledge my team, the Applied Weed Physiology Lab at Penn State, to my committee that are providing insights constantly, and also to the funding agency of this program. Thank you very much.
I'm sure Adrian would like to answer questions.
Yes.
Hi, Adrian. That was interesting. When you showed your slide with the different mechanisms by population, I was trying to imagine a map in my head, and I'm wondering if you looked at the mechanism type by distance, like geographic distance.
Yes, I have a plot where I didn't add it here, but Most of the cases are CMV driven. I also conducted a PCA. The PCA also it's mostly explained by the type of resistant mechanism. And the isolation by distance analysis is giving me a slight positive correlation between the distance and the populations. But so your question comes, maybe are you asking if there might be gene flow spreading the mechanisms?
Yeah, and like yeah, so whether the populations that are closer together are like showing more similar mechanisms because of that.
Yes. For instance, Pennsylvania, New York, and Maryland. No, Maryland it's a non-target type, but Pennsylvania, New York, they have the same mechanism. It's CMV. We have populations from South and North Carolina. They're also CMV. We have a lot of populations in Oregon. They're also CMV. But then I also conducted a admixture analysis, and that it's kind of giving me a lot of admixture across populations, except for the South Carolina and North Carolina. It seems like something is happening, something more related is happening between those populations. But then again, the H12, which is a method to infer the haplotype diversity carrying the derived alleles, it's showing me that there's a lot of variation in haplotypes. So maybe that I still need work to do to infer better what is happening, but it seems that it might be a case of parallel or repeated evolution in different cases.
Cool. Okay. Anybody else, or does Robin want to ask another question since you're the weed specialist here? I have a question. Do you find doing the fieldwork gives you ideas about what to do on the genomics level?
Sorry, can you repeat your question?
Do you find being out in the field and looking at where the weeds are, where the Italian barley is?
Okay, yeah. Well, actually, the the seed collection was just collaborators from my PI that shared the seeds, and then we did everything in the greenhouse. So I didn't have the chance to go out and, I don't know, take some information from outside. But yeah, that would have been nice. But I'm mostly greenhouse lab and then in front of the computer.
But that is realistic, but I still think it's useful to do a little fieldwork, even if you're going to do most things in a common environment, just because it might give you some ideas. But it was very cool. I really enjoyed your talk.
Thank
All
you.
right. How much more time do we have, Briley?
We don't have another speaker, so Adrian was our last
where are
one.
we? We're over time, right? We're at
Yes.
yes. Okay. Well, I want to thank Riley for being such a very good technician, and I really enjoyed everybody's talks. And yes, hopefully I'll see you either next year at the Virtual Evolution meetings or maybe in person. Certainly, some people are going to be in person. So goodbye. I think this was a particularly good session.
Have a nice weekend. Bye.
Thanks, Elizabeth,
Yeah,
for
thank you. You too. See you in Cleveland.
Riley for
If you're
having
going.